Hello students, welcome to my lecture series. Myself Dr. Sangita Bhamare, Department of Zoology, S.V.K.T College, Devlali Camp, Nasik. In my last lecture, we saw the structure of cell in which prokaryotic cell and eukaryotic cell. Means in last lecture we saw what is mean by cell, what is cell biology, significance or applications of cell biology in various fields, and next to it we saw the detailed structure of prokaryotic cell and eukaryotic cell. Today I am going to talk about this plasma membrane, which is the separate chapter as which is mentioned in our syllabus. So in this lecture I am going to talk what is plasma membrane, structure of plasma membrane, composition of plasma membrane, what is fluid mosaic model, and what are the functions of plasma membrane. So basically, first of all, what we can understand with this particular chapter or regarding this plasma membrane. So here we will get the detailed information regarding this plasma membrane, what is its ultrastructure, how it works in prokaryotic cell and in eukaryotic cell. So these are the learning objectives behind this chapter, plasma membrane. So before going to start this plasma membrane, I would like to tell you the difference between prokaryotic cell and eukaryotic cell. So these are the pictures. Towards the left you can see this prokaryotic cell and right indicates the structure of eukaryotic cell. So first difference in between this prokaryotic and eukaryotic cell. So regarding the cell size, so cell size is in case of prokaryote 0.2 micrometer to 2 micrometer diameter, while in eukaryotes it ranges from 10 micrometer to 100 micrometer. So what's about the cell wall? Usually cell wall is present in prokaryotes and chemically complex in nature as compared to eukaryotes. So what happened in eukaryotes? Only in plant cell the structure of cell wall is present, and we saw in our lecture that it is made up of the cellulose, while in animal cell as such the cell wall structure is totally absent. So what is nucleus? So in case of prokaryotes rather than nucleus, here the structure which is present is known as a nucleoid, basically which is without nuclear membrane. That's why we say that true nucleus is absent in prokaryotes, while the name itself indicates eukaryotes means the true nucleus, right? So here in case of eukaryotes the true nucleus is present. What's about the ribosome, which are one of the important subcellular organelle? So what happened in prokaryotes, the ribosome structure which are small and 70S type of ribosomes are there, while in case of eukaryotes they are present and comparatively larger in size, we call it as a 80S type of ribosomes. So as per the sedimentation rate here 70S and 80S, these labels are given to this particular structure of subcellular organelle, that is ribosome. So DNA arrangement if we are talking about, so in case of prokaryotes the DNA is circular double stranded, while in eukaryote the structure of DNA is totally linear. Then mitochondria. As such, this subcellular organelle is totally absent in case of prokaryotes, while well-developed double membrane structure of mitochondria can be seen in eukaryotic cell. What's about this cytoplasm? Cytoplasm is present where subcellular organelles are absent. So what happened? We can't say as such the subcellular organelles are absent in case of prokaryote. But what happened here? Very few subcellular organelles are present in prokaryotes, while well-developed membranous subcellular organelles are present in case of eukaryotes. Then next subcellular organelle which are enlisted here like endoplasmic reticulum. So endoplasmic reticulum is absent in case of prokaryotes, while endoplasmic reticulum is present, network-like structure, in case of this eukaryotic cell. Then plasmid. Plasmid, which we called it as an extra chromosomal material, which is present in case of prokaryote, but as such this structure is not present in case of eukaryotes. Then ribosome. We saw ribosomes are small, then 70S, while in case of eukaryote they are comparatively larger in size and 80S type of ribosomes are there. Then lysosomes and centrosomes are totally absent in case of prokaryotes, while lysosomes which are formed or synthesized with the help of Golgi bodies These structures are present in case of eukaryotes. So lysosome and centrosome, these structures are present in eukaryotes, and in prokaryotes these are absent. Then cell division. How cell division takes place in prokaryotes? So basically cell divides by binary fission in case of prokaryotes, while in case of eukaryotes the well developed cell division mechanism can be seen. We called it as a mitosis. Then flagella, which is one of the locomotory organ. So these flagella are almost similar in size in all prokaryotes, but here the flagella are larger or quite different kind of structure can be seen. Then if you are talking about the reproduction, so asexual reproduction is there in case of prokaryotes, while in case of eukaryote both sexual as well as asexual reproduction can be seen. Then good example for the prokaryotes we have already seen, that is E. coli or bacteria, some archaea, and for eukaryotes you can take the example of typical plant cell or animal cell. So this is all about the differences, detailed differences in between prokaryotic cell and eukaryotic cell. Now we turn towards this chapter, plasma membrane. So what is plasma membrane? First of all, if you want to define this term plasma membrane, how you can define it? So plasma membrane which encloses every type of cell, both prokaryotic cell as well as eukaryotic cell. So basically it is responsible to separate the cytoplasm from the surrounding cellular environment. So the first role of the plasma membrane is it is responsible to separate the cell, its cytoplasmic material from the outermost environment. So this is the structure of plasma membrane or definition of plasma membrane. So here you can see some features related to this plasma membrane. So what are the features you can see related to this plasma membrane? So basically it's the ultra thin and elastic, leaving dynamic and selective transport barrier. So as per the transportation mechanism, it is able to change itself. That's why it is called as a dynamic structure. It's leaving, if you're talking about in vivo condition, it shows the elasticity, it is very thin, and finally we can say it's the selective transport barrier. Then it's the fluid mosaic assembly of molecules as lipids, in which you can take this phospholipids and cholesterol, which is the actual composition, then proteins and carbohydrates. So in next slide, I'm going to tell you which type of lipids are there, which type of proteins are there, and which type of carbohydrates are there. Then if you're talking about the occurrence, means where it is present actually. So the cells of bacteria, plants as well as animals. So in all this kind of organisms, you can see the well-developed structure of plasma membrane where it works as an envelope and selective transport barrier. Then here functions of plasma membrane. So what is the role of plasma membrane? First, plasma membrane controls the entry of nutrients and exit of waste products and generates differences in ion concentration between interior and exterior of the cell. So basically all the essential material, minerals, nutrients will be transported inside the cytoplasm through this structure called as a plasma membrane, as well as same structure work to transport this waste material which is suppose present inside the cytoplasm. Next, it has been observed that this plasma membrane also helps to maintain the concentration of the minerals inside the cell as well as outside the cell. Then it also acts as the sensor of exterior signals, means accordingly it is ready to give the particular action against the particular signal. So that signal may be hormonal type of signal, then or immunological signal or neurological signal, etc. So if you are talking about some other properties of this plasma membrane, so all biological membranes including the plasma membrane and internal membrane of eukaryotic cell, that is membranes bounding endoplasmic reticulum, then this nucleus, mitochondria, chloroplast, Golgi body, lysosome, peroxisome. So basically when we talk about this subcellular organelle, so how we define this subcellular organelle? So these are the membrane bound subcellular organelle. Now if you want to compare the structure of plasma membrane which is present outside this cell and the structure and composition of the membrane which is present in the subcellular organelle. So all these structures are almost same, only few these biomolecules as carbohydrate, proteins and lipids, their concentration is different. So otherwise this membrane structure is same in plasma membrane of the cell as well as in the membrane bound subcellular organelle. Now we are going to talk about the structure of this plasma membrane. So basically structure of plasma membrane, when we define, when we describe, then it has been observed that this structure is made up of this lipid bilayer structure. So this lipid bilayer structure is firstly discovered by Singer and Nicholson in 1972. So many hypotheses and theories which are associated with this structure of plasma membrane, but the well accepted theory is the theory which was put forward by Singer and Nicholson in 1972. So basically, according to Singer and Nicholson, the structure of plasma membrane is labeled as fluid mosaic model. So why it is fluid mosaic model, that I will tell you later on. But before that, in this picture, what you can see basically the yellow color structure is the structure of lipid bilayer. So majority of which type of lipids are there? Here phospholipids, by which this lipid bilayer structure is made by. Then this blue color structure indicate the type of protein. So here two types of proteins, you can see channel proteins are there and this integral proteins are there, peripheral proteins are there. So two types of proteins, this integral proteins and peripheral protein, which are then divided into this various type of protein. So basically here some globular proteins are also there which work as a channel protein. Then glycoproteins are there which are nothing but the carbohydrate plus proteins. Glycolipids means carbohydrate plus lipid. Then few little or trace amount of cholesterol also can be seen. So what is the role of each and every biomolecule which is supposed present in this structure, that we will see later on. So this structure is called as a fluid mosaic model and which was suggested or discovered by Singer and Nicholson in 1972. So question arises, what is fluid mosaic model or why this structure is labeled as a fluid mosaic model? So fluidity, it is according to the nature of the fluid or water, etc. Then it shows this membrane permeability and cholesterol. So when we look towards the property of the water, so what is the property of the water? It works for the transportation. So same type of function is carried out by this fluid mosaic model or this lipid bilayer structure. That's why we called it as a fluid. So it works like a fluid. It shows the property like a fluid. That's why we called it as a fluid model. Then what is this asymmetry? So asymmetry, this is one of the important property which we can see in this previous structure of this fluid mosaic model. So basically due to the arrangement of the proteins in the structure of this lipid bilayer, here the structure as such is not the symmetrical structure. So rather than symmetry, here we people can see total asymmetry. That's why we call this structure as a asymmetric structure due to arrangement of proteins, both peripheral as well as this integral. The next is mosaic structure. So here if you observe carefully, the structure of proteins which are embedded are just like your mosaic tiles. That's why we call the structure as a mosaic model. So this is all about the properties which are associated with this particular label, that is fluid mosaic model. Then we turn towards the composition which is associated with the structure of plasma membrane. So first composition is the lipids. So four major classes of lipid. Remember here one thing, majority of this type of biomolecules are present in the structure of fluid mosaic model or in plasma membrane. So that biomolecules are the lipids. In lipids again, it has been observed that four different type of lipids are there. So which are that four different type of lipids? So first major are the phospholipids, then some sphingolipids are there, glycolipids and very few sterols, as we saw cholesterol is there. Then all of them are amphipathic molecule. This is one of the property which is associated with this lipid molecule. Amphipathic means having both ends, hydrophilic as well as hydrophobic. So this structure shows, lipid biomolecule structure shows both domains as hydrophilic one and hydrophobic one. So basically it is called as, if you observe the structure of this lipid biomolecule, single lipid molecule. So here the head or button-like structure you can see, which is polar or hydrophilic in nature, and hydrophobic long fatty acid chain is there, which is called as a hydrophobic tail. So the relative proportion of this lipid, which varies in different membranes, as I already told you, that this membrane structure, if you observe in plasma membrane of the cell and in membrane bound subcellular organelles, then more or less the composition or content is same, only the proportion is different. So phospholipids may be acidic phospholipids near about 20%, such as sphingomyelin, neutral phospholipids, and phosphatidylcholine and phosphatidylserine. So basically 3 different type of complex phospholipids can be seen in this plasma membrane as phosphatidylcholine, phosphatidylethanolamine, and phosphatidylserine. Many membranes contain this cholesterol also. So question arises because this cholesterol, so many questions which are in our mind related to this cholesterol. So what is the role of this cholesterol? Cholesterol is especially abundant in the plasma membrane of all mammalian cells and totally absent in case of prokaryotic cell. So what is the role of cholesterol? Remember here, it helps to maintain the membrane permeability. So what is mean by membrane permeability? So membrane permeability is nothing but the passive diffusion of the molecules which depends upon the electric charge and polarity of that particular content or molecule. Then next type of lipids, cardiolipin. Remember here, cardiolipin especially are present in the inner membrane of mitochondria rather than the structure of plasma membrane. So this is all about the lipid content which is present in the plasma membrane. The next, proteins. The protein or the amount of type of protein in the plasma membrane are variable. So in the myelin, which is associated with the nervous system, which serve plasma membrane, contain about 50% protein. So mainly to insulate the nerve cell axon, less than 25% of the membrane mass is protein. So majority of proteins which are present, if you are talking about this membrane and all this, it is associated with the myelin chain. While in the membranes involved in the energy transduction mechanism, as in the mitochondrial inner membrane, chloroplast inner membrane, etc. So here 75% of the proteins are present which help in the mechanism of this energy transduction or the synthesis of ATP molecules, so other high energy components, etc. According to their position in the plasma membrane, so basically 2 types of proteins are there. Integral protein, which are also known as the intrinsic protein, and peripheral or extrinsic protein. So as per the location, these proteins are labeled. The intrinsic proteins tend to associate firmly with the membrane or this lipid bilayer. So remember the type of interactions which are working in this integral protein and lipid bilayer are the hydrophobic interaction, while the association which we can see in between the extrinsic protein and this lipid membrane is the electrostatic association or electrostatic interaction. So question arises, what are these interactions? So remember here, this membrane stability is maintained according to, or it depends upon the type of interactions which are working here. For example, hydrophobic interaction, electrostatic interaction, etc. So here some examples which are associated with these proteins are given, basically are present in this membrane. So some structural proteins are there, then enzyme transport proteins are there like permeases or carriers. So later I will show you which type of enzyme transport proteins are working in this particular plasma membrane. Some of them may act as antigens or receptor molecules. So when we go through the detailed structure of this plasma membrane, so with the composition here, few proteins which are present in the structure of this plasma membrane and so many functions which are associated with this protein. So from this, some proteins are structural proteins, some proteins are enzyme proteins, some protein work as a receptor molecule for various hormones. So this is all about the variety of types of proteins which are associated with the structure of plasma membrane. Then next to lipids and proteins, we turn towards the carbohydrates. So carbohydrates are present only in the plasma membrane. They are present as a short, sometimes branched, sometimes unbranched in the form of small or long oligosaccharide chain. So if you observe carefully, the structure of fluid mosaic model, in the structure of fluid mosaic model, some glycoproteins recently we saw, then some glycolipids are there, etc. So here, this small oligosaccharide chain can be seen in association with the peptides as well as some fatty acid chain. So according to some viscosity experiment, they suggest that the structure of plasma membrane is not so similar with the oil, but it is too much similar with the other fluids or liquids like water. And due to this means the arrangement or composition of the lipids and proteins, it shows the particular movement. So that movement is called as a flip-flop movement. So that flip-flop movement which is in between this lipid and protein molecule. So because of this flip-flop movement, it is able to transport some material from exterior to interior or from interior to the exterior side. So basically transportation of the material is possible because of this particular movement of the lipid bilayer or this lipids or proteins, etc. So that movement is called as a flip-flop movement. So on account of its fluidity and the mosaic arrangement of protein molecule, this model of the membrane structure is known as the fluid mosaic model. So that we have already seen in previous slide that due to the nature, due to the function or arrangement of lipids and proteins which is associated basically with this lipid bilayer, this structure is called as a fluid mosaic model, and the structure which was suggested firstly by Singer and Nicholson in 1975. So the lipid layer which is defined as a bimolecular and the protein is extended, but the different on two faces of the protein that we have already seen, means the structure by which this lipid bilayer is made by the hydrophobic tail of the lipid protein and hydrophilic head of this particular structure which is polar in nature. Then transportation. Transportation is the important function which is carried out by this lipid bilayer or structure of plasma membrane. So which type of transportation? So many ways of transport mechanisms are there. From this, we are going to start this with the active and co-transport mechanism. So what is active transport mechanism? So in active transport, a solute is moved against a concentration or electrochemical gradient. Means remember, active transportation is possible only in that situation whenever there is a electrochemical gradient is there. So it helps to transport the proteins, consume the metabolic energy, usually ATP. So with the help of ATP molecule which acts as a high energy bond, here the solute will be transported accordingly, means towards the exterior side or towards the interior side. So in primary active transport, basically the enzyme, one of the globular protein or channel protein which is present inside this plasma membrane which helps, that protein is called as a ATPase enzyme. So how it works, that we people can see in this sodium potassium ATPase pump. Remember, sodium potassium ATPase pump is one of the type of active primary transport mechanism. So in this picture you can see this sodium potassium ATPase. So I am going to describe how this transportation mechanism works. So basically here that greenish structure indicates the lipid bilayer structure and in which this integral proteins can be seen, channel proteins can be seen called as a ATPase. So remember, ATPase are one of the type of a channel protein and name itself indicate it may be one of the enzyme. So ATPase which is one of the globular protein or enzyme. Here it helps for the mechanism of transportation, especially the minerals like sodium and potassium. So how it works? So basically, if you are going to talk about the structure of this ATPase molecule, so recently I told you it is one of the type of globular protein. So it is made up of globular protein, or it is made up of different type of globes as 2 alpha chains, 2 beta chains and 2 gamma chains, right, where each subunit or each chain is responsible for the particular function. Now next, how it works? So ATPase, which is one of the type of channel protein, so it helps to maintain the mineral balance. Which mineral balance? I told you, sodium and potassium. So fixed concentration can be seen in this particular picture. So what is that concentration? Which is in millimolar form. So towards the extracellular, always sodium should be near about 150 millimolar, and always this potassium should be 5 millimolar. While inside the cell or towards the interior side, the sodium should be only 30 millimolar and potassium should be towards 150 millimolar. So this is called as a electrochemical balance. So this balance is always possible only because of this pump called as a sodium potassium ATPase pump. So how this ATPase enzyme works here? So role of ATPase, as I told you, it is one of the type of channel protein known as. So it helps to degrade the structure of ATP molecule. So what is ATP? Adenosine triphosphate, which is one of the type of high energy molecule with a high energy bond. So after degradation or hydrolysis of the structure of ATP, what happened here? It will be converted into ADP and one inorganic phosphate. Then suppose after degradation we can see ADP and one inorganic phosphate is going to form. But what is the use of this degradation or hydrolysis? So remember, after degradation or after the breaking of that particular phosphate bond, here large amount of energy is released, and that energy is utilized to transport this 3 sodiums towards the exterior side, and at the same time this 2 potassium inside the cell. So this is how this sodium potassium ATPase pump works, because of which the electrochemical balance can be maintained inside the cell. So this is all about this today's lecture, plasma membrane. So what we saw in this lecture, we saw the outermost covering or envelope, which is nothing but the plasma membrane. So how we define this plasma membrane? Definition of plasma membrane, so which is the outermost covering or outermost envelope. Then as per the location here, what is the function of plasma membrane? So with the protection, which is the outermost covering, here another important function which is carried out by this plasma membrane is the transportation. Recently we saw here that example, that is active transport mechanism or sodium potassium ATPase pump. Then we saw the composition. So here plasma membrane is made up of this important biomolecules, lipids, proteins and carbohydrate. Then structure of plasma membrane. In the structure of plasma membrane, this lipid bilayer, we saw what is that lipid bilayer, its composition, and what is this fluid mosaic model. Few questions which are associated with this today's lecture that you have to solve and upload it on our Google Classroom. So first question is, what is plasma membrane? You have to define it as well as briefly you have to explain it. Then next question is, what is primary active transport? So here you have to draw the neat label diagram of this sodium potassium ATPase pump, and then next you have to explain in that particular pump what is the role of this ATPase enzyme. This is all about the today's lecture. In next lecture, we are going to talk about the detailed mechanism of secondary active transport, which is associated with the transportation mechanism, and the next or another transportation mechanisms, which are endocytosis and exocytosis, then diffusion, what is facilitated diffusion, what is symport, uniport and antiport. So all these mechanisms which are associated with the transportation, we will see in next lecture. Thank you.